So what happens if you get to the end, but your problems have just started? Hi biology friends, it's Sarah, your other biology professor, and let's talk about telomeres and the end replication problem. Because eukaryotes have a bit of a problem when it comes to DNA replication, and that's because our chromosomes are linear. They've got ends, and with the way that DNA replicates, that means that chromosomes can shorten over time. To understand why this happens, we need to remember a few important things about DNA replication. Of course, you know that each existing DNA strand will be used as a template to build a new complementary strand. But because DNA strands are anti-parallel, opposite 5' and 3' directionality, and because new nucleotides are added in the 5' to 3' direction only, so adding on to the 3' end of an existing strand, that means that the new strands are built in different ways. And so we end up with a leading strand synthesized continuously and a lagging strand synthesized in fragments that are joined together. Okay, so why is that a problem at the ends of the chromosome? Well, think about how each of those fragments is started. With a primer, an RNA primer, that'll be removed and replaced with DNA. The RNA primers are necessary because DNA polymerases can't start a strand from scratch. They need a 3' hydroxyl, OH, to add new nucleotides onto. Now, RNA polymerases don't have this problem. That's why you don't see primers used in transcription. And so an RNA polymerase that you may know as primase will throw down some RNA primers that'll give DNA polymerase something to build off of. And so in eukaryotes with multiple origins of replication, multiple replication bubbles will arise, and the elongated strand from the adjacent replication bubble will provide that 3' hydroxyl group needed to fill in the gap after the internal primers are removed. The problem comes with the terminal primers, the ones on the end of the lagging strand. When those are removed, there's no upstream piece of DNA for DNA polymerase to build off of. It has no 3' hydroxyl group. And so those gaps, they stay. This isn't a problem for circular chromosomes, no ends. So replication around that circle will leave you with a nice, fresh 3' OH to build off of in order to fill that gap. But with linear chromosomes, we're stuck with that empty space. So when that DNA molecule is copied and that shortened strand is used as a template strand, the new double-stranded DNA molecule will be shorter. Genetic information is lost. And every time the DNA replicates, the gaps from the terminal primers remain unfilled, and the new DNA molecule shortens even more. And that's the end replication problem. But some types of cells have solved this problem. How? Telomere replication. At both ends of eukaryotic chromosomes, you'll find telomeres. Telomeres are regions of highly repetitive sequence. In humans and other vertebrates, that sequence is TTAGGG. And you'll see that repeated potentially thousands of times at the ends of the chromosomes. So no genes here. And that's handy because as cells keep dividing, DNA is replicated, chromosomes are shortening, shorten too much, and eventually you'd be losing genes. You'd be losing very important genetic information. So instead, it's repeats on the ends. Now, much of the telomere DNA is double-stranded, but you'll find a 3' overhang on the very end, the result of primer removal. You'll also see this called the G overhang because the sequence of the strand that it's on is guanine rich, lots of Gs. And it's not just the DNA sequence that's important here. Just like the rest of the chromosome, at the telomeres, you'll find proteins interacting with DNA. But here, it's not just the histones that you already know. Instead, you'll find other, more specialized proteins that are doing things like protecting the chromosome ends from degradation and making them not look like double-stranded breaks that need to be repaired. And these proteins can also help to control how long the telomeres are by regulating the activity of a protein called telomerase. Telomerase is a ribonucleoprotein enzyme That means it's a complex of protein and RNA. It's also a special reverse transcriptase. That means that it makes DNA from an RNA template, the reverse of transcription. So telomerase brings its own RNA molecule with a sequence that's complementary to the telomere sequence. And it binds in a sort of offset manner with that 3' overhang. Telomerase uses its own RNA sequence as a template to extend that 3' overhang. And then it translocates, it moves, and extends that overhang even more. Through a process we're still working out, a complex of proteins, including DNA polymerase alpha, primase, and single-stranded telomere binding proteins, work to fill in the DNA missing on the other strand. The RNA primer is removed, the gap remains, but we've made the telomere longer. Perfect, problem solved, right? Well, telomerase isn't active in most adult human somatic cells. The gene that encodes the protein just isn't transcribed. Bummer. Most human cells have a limit on the number of times they divide before they stop dividing. It's called the Hayflick limit, and it's correlated to telomere length. A human telomere may lose 40 to 200 base pairs every time the cell divides. And when the telomeres get too short, the cell either dies or it stops dividing and enters senescence. When cells age, the organism ages. But is a short telomere the cause of aging or just another symptom of aging? We're not sure yet. Many scientists believe that other types of DNA damage that accumulate over time might be a better place to look. I said that most human cell types are limited in their number of divisions, but what about the others? Cell types that are highly proliferative, meaning they have to divide a lot in order to do their job, will have active telomerase to help maintain their telomeres and get around that limit. This allows them to keep dividing. So things like germ cells, the cells that divide to produce sperm and egg, certain types of stem cells, epithelial cells, and cells in the immune system as well. Cancer cells are able to get around this limit by reactivating telomerase, allowing them to divide uncontrollably. Upwards of 85% of human cancers show active telomerase, and the ones that don't seem to have acquired alternative mechanisms for preserving their telomeres, allowing them to essentially be immortal. And so, of course, a lot of research on aging and cancer is interested in telomeres. Could we reactivate telomerase and preserve telomeres in order to prevent aging? Could we do that without promoting cancer? Or could we turn off telomerase in cancer cells as a form of treatment? We'll see. There's good stuff at the end of your chromosomes. If you learned a little something about telomeres, like why they're so important but tricky to replicate, give me a thumbs up. Be sure to subscribe for even more biology content from me. Check out my website for tutoring resources, and I will see you next time. Bye.